Hi everyone. My name is Dr. Jang, who is an assistant professor in animal and Department of Animal and Food Science at University of Wisconsin River Falls. Firstly, thanks to Chungang University for inviting me in this seminar. And today's topic is extra-phosphoric effects of phytase in swine diet beyond the phosphorus release. So now let's get started. Phytate is a storage form of phosphorus found in cereal grains and oilseed meals. So as we are using corn and soybean meal in high amount in swine diets, we always have this phytate in the swine diet. And the accumulation site is alluron layers. So in this alluron layer of cereal grain and oilseed, we can find not only protein, but also we can find this phytate and other fibers. And because this phytate binds to phosphate group, 6-phosphate group here, it reduces phosphorus availability. And also it can bind to calcium, zinc, iron, fat, protein, and starch. So phytate can reduce not only phosphorus availability, but also the other nutrient utilization in swine diets. So this phytate with this property, it has anti-nutrient effects. So firstly, it inhibits endogenous enzyme activity, like such as pepsin. So high content of phytate reduce pepsin activity and other digestive enzymes. So it increases endogenous loss of amino acid and minerals because it can bind to amino acid and minerals and other nutrients. So then it increases endogenous secretion in gastrointestinal tract of pigs. So in order to reduce this, in order to improve phosphorus availability and reduce anti-nutrient effect of phytate, we use phytase enzyme. So this phytase enzyme sequentially remove phosphate group, okay, in this phytate and liberate phosphate from phytate so that excuse me, pigs can use this phosphorus in these cereal grains and oilseed meals better than without phytase in the diet. So this phytase supplementation effect is very clear in improving phosphorus digestibility. However, because this phytate can bind to other minerals and other amino acid and energy yielding nutrients, etc., when you use phytase in the swine diet, it could improve macro minerals and trace mineral availability and digestibility and availability of amino acid and energy. And this is called extra-phosphoric effects, which is a further response of pigs to phytase supplementation beyond phosphorus release. And then we could expect this extra phosphoric effect by using phytase in the swine diet at high level. That is called superdosing phytase. So this is using phytase enzyme at over 500 FTU per kilogram diet, which is typical level of phytase in the swine diet. You could use 1,000, 2,000 FTU of phytase per kilogram diet. That is superdosing phytase. Then the purpose of this superdosing phytase supplementation is completely degrade or phytate in the diet, okay, and then so that we could remove anti-nutrient effect of phytate. So then we can expect extra phosphoric effects from enhancing micro minerals availability and the other nutrient utilization. So this extra phosphoric effect phytase potentially, okay, can be potentially found in sows and growing and finishing pigs, so or the or pigs. So firstly, in sow, when you use phytase and probably superdose level, you could expect this extra phosphoric effect in weight loss of sows in lactation and their litter performance, and probably milk phosphorus content and bone integrity of their progeny. And if you use the phytase in growing and finishing pigs diet, extra phosphoric effect could be in growth and feed efficiency, nutrient digestibility, and carcass characteristics. So in this seminar, we will look at several research results that demonstrated these extra phosphoric effects of phytase in sows and growing finishing pigs. So firstly about sows. So this first research result have two experiments. So first one is multi-state study with University of Georgia, University of Arkansas, and North Carolina State University. And second experiment was conducted at University of Kentucky with phytase supplementation on to the sow diet on reproductive performance and apparent total tract digestibility and bone characteristics. In experiment one, with 3 universities involved, we used 365 liters from 182 sows, and we had 4 treatments in this study, and phytase was supplemented in gestation and lactation diet from breeding. So negative control diet was low calcium and available phosphorus diet, and positive control was normal calcium and available phosphorus diet. And then we added 300 and 600 FTU of phytase per kilogram diet in negative control diet. And then in second study, we used 16 sows and we supplemented phytase to the gestation and lactation diet from day 32 to 52 of gestation. And as negative control diet, uh, uh, we used low calcium and phosphorus diet, and we had positive control diet, no more calcium and available phosphorus diet, and we added 500 FTU of phytase in low calcium and available phosphorus diet. And in experiment one, we measured sow and litter performance. Then as you can see in this table, sow body weight and weaning to estrus interval and lactation feed intake uh, was not different by dietary phytase supplementation. However, in litter size, the number of total born and live born increased quadratically as phytase supplementation increased from 0 to 600 FTU. So 300 FTU per kilogram treatment has the highest total born and live born numbers. And this is maintained numerically in number of winning pigs. So uh, even though the effect of phytase on the number of on the litter size is not clear, uh, we found this improvement in litter size, and some previous research also showed similar result. And if you look at litter performance, litter weight of live born increased, okay, and peaked at 300 FTU of phytase per kilogram diet, and without any difference in piglet body weight of live born, this result may be from increased litter size. However, piglet winning weight increased linearly, okay, with increasing phytase supplementation level up to 600 FTU, and this means phytase affected okay, sow, probably milk and others, so that the piglet growth increased with increasing phytase supplementation level. So then in experiment 2, we measured phosphorus and calcium digestibility of sows in gestation and lactation. Firstly, phosphorus digestibility, we could see a clear effect of phytase in increasing phosphorus digestibility in both gestation and lactation period. So in gestation period, phosphorus digestibility increased, and it is comparable to positive control diet, normal calcium and phosphorus diet. And in lactation, phosphorus digestibility is also increased, but it was over normal calcium and phosphorus diet. So this result means phytase supplementation in gestation and lactation diet improve phosphorus digestibility clearly, and in lactation period, there is more pronounced effect of phytase supplementation compared to gestation period. And it is because lactation period sow requires more nutrients, okay, so that we could expect more pronounced effect of this phytase supplementation in lactation period. And calcium digestibility here, uh, uh, the in gestation period, there was no improvement detected in calcium digestibility, but lactation period, calcium digestibility is increased, okay, compared to increased here, okay, increased compared to no phytase diet. So this means phytase supplementation not only increase phosphorus digestibility, but also other minerals, here calcium digestibility. And actually, phytase supplementation effect on phosphorus digestibility in gestation period is inconsistent. And in this table showed several previous research result, including this study, and it contains collection period and whether there was phytase effect or not, and then phytase level and phosphorus content in the control diet. And when the study detected positive phytase supplementation effect in increasing phosphorus digestibility, they had low phosphorus content in the control diet, or it has late gestation period as a fecal collection period. And then when there was no phytase effect detected, it had high phosphorus content that meets requirement, and then their fecal collection period is mid gestation period. So phytase supplementation effect in gestation period is depending on total phosphorus content in the control diet and then period of fecal collection. So late gestation period, due to fast fetal growth, the sows requires more nutrient. So we could detect more pronounced phytase effect. And then phytase effect is also feasible when you use low phosphorus content in the control diet, okay, compared to when you have high phosphorus content, normal phosphorus content in the diet. And then in experiment 2, we also measured bone characteristics of sows and piglets. So sow bone ash content was not different by adding phytase in the in gestation and lactation diet, but piglet bone ash content and piglet bone strength, okay, at birth, there was no significant difference, but at weaning, bone ash content increased. Okay, increased when you supplement diet with 500 FTU of phytase compared to no phytase supplementation, and bone strength as well. Bone strength increased, okay, when you supplemented diet with 500 FTU of phytase. And this result might be attributed to phytase effect on increasing milk phosphorus content. Unfortunately, we did not measure milk phosphorus content because we did not collect milk sample. But in previous study on 2011, they used 500 FTU, okay, phytase in this MP and EP treatment, okay, with other enzymes in EP treatment. Uh, then in gestation and lactation period, phytase supplementation increased milk phosphorus content, okay, compared to negative control diet. So supplementing phytase in sow diet, okay, in gestation and lactation, the diet could increase phosphorus content in milk, and then this may affected, okay, milk piglets bone ash and bone strength uh, because they are drinking this milk. The next study is about superdosing phytase supplementation to a lactation diet conducted at University of Kentucky. And we used 17 sows in this study with corn soybean meal based diet, and we added 0 and 3,000 FTU of phytase per kilogram diet and only in lactation period. And we measured standard reproductive performance and parental tract digestibility and milk composition and phosphorus content. Firstly, there was no statistical difference by superdosing phytase supplementation in lactating sow diet on sow body weight and lactation feed intake. And then there was no significant difference in litter size and piglet weight and litter weight in this study. Also, when we measure milk composition for fat, protein, lactose, total solid, and solid not fat, there was no significant difference. And then milk mineral content for phosphorus, zinc, calcium, and manganese in early and late lactation period, there was no difference. And in previous slide, we looked at milk phosphorus content increased by supplementing phytase in gestation and lactation diet. So the discrepancy between these two studies indicated that uh, in order uh, in order to increase phosphorus content in the milk, we may need a longer time period of supplementing phytase in sows. Okay, so only lactation period, it may not be enough, okay, to increase milk phosphorus content. And then, however, superdosing phytase supplementation in lactation diet reduced weaning to estrus interval from 5.7 days to 4.0 days, which means there is some positive effect of superdosing phytase supplementation in lactation diet. And it's not clear, okay, the mode of action is not clear, but phytase supplementation could increase phosphorus digestibility and other nutrient digestibility, and also the reduced anti-nutrient effect of phytate, so it could affect sow health condition, okay, so that it could reduce weaning to estrus interval. So summary of these sow studies here, firstly, phytase supplementation to gestation and lactation diet improved the phosphorus and calcium digestibility, and we found lactation has more pronounced effect of this phytase supplementation. and then it could increase weaning weight of piglets and increased bone ash content and bone strength of their progeny. And superdosing phytase supplementation in lactation diet did not increase milk phosphorus content, then because it may be short duration of supplementation of superdosing phytase, but it reduced weaning to estrus interval. But we need further study to clearly demonstrate the superdosing phytase effect and their extra phosphorus effect in sows. And next study is for growing and finishing pigs using both Xylanase enzyme and Phytase enzyme to improve nutrient utilization and feed efficiency. So as we are using cereal grains and oilseed meals in swine diets, there are always indigestible portions in swine diet, which is mainly fibers and phytate. And then if you use wheat sources, wheat byproducts, and some fibrous byproducts, you could find albinogyllans and phytate in those ingredients. So in this table it shows albinogyllans and phytate phosphorus content in different feed ingredients, mainly used in swine diet. So corn and soybean meal, there is albinogyllans and phytate phosphorus. But we also use fibrous byproducts to reduce feed cost for growing and finishing pigs because they usually have better ability to use fiber compared to younger age pigs. But then if you use these fibrous byproducts such as corn germ meal, wheat middling, and corn DDGS, you can find that there is more fiber and more phytate. So a lot higher level of albinogyllans and phytate phosphorus in these byproduct. So increased use of this byproduct increase the content of the level of albinogyllans and phytate in the diet. That means there is more indigestible portion in growing and finishing pigs diet. Then, in order to increase these nutrient availability, we use Xylanase and Phytase enzymes in the swine diet. So then, we could expect synergistic effect between Xylanase and Phytase supplementation because in aileron layer of oilseed and cereal grains, you could find both albinogyllans and phytate. So then, when you supplement both Xylanase and Phytase in the swine diet, Xylanase can break down this fiber, albinogyllin, in aileron layer, okay, so that it increases permeability of this aileron layer, which is also the site of phytate storage. So then, it could improve accessibility of phytase to the phytate so that it can improve the action of phytase to the phytate. Okay, so we could expect synergistic effect of this between Xylanase and phytase in swine diet. So in this study, we used corn DDGS and wheat middlings and corn germ meal as fibrous byproducts to provide enough substrate for Xylanase and phytase. And then we used graded level of phytase supplementation up to superdose level, which is 0 to 2000 FTU per kilogram diet. And we used Xylanase in the diet or not. And we measured gross performance, carcass characteristics, and apparent total direct digestibility of pigs. So we used 45 individually fed pigs with initial body weight 26 kilogram and have 9 treatment in 5 replicates. So one positive control diet have high energy, and then negative control diet has low energy content compared to positive control diet. And then we had 2x4 factorial arrangement with first main factor phytase level 500, 1000, and 2000 FTU per kilogram diet. and then Xylanase supplementation without Xylanase supplementation, or 24,000 BXU Xylanase per kilogram diet supplementation. So this table shows you the treatment structure. So positive control, negative control, no phytase and Xylanase. And then we have phytase diet, and then both phytase and Xylanase diet, so that we could detect the interaction between phytase and Xylanase if there is any. We used the four-phase feeding program at Lipton feeding, okay, with free access to water, and then 0.3% titanium dioxide is used for phase 4 diet for apparent total tract digestibility calculation. So we collected feces for digestibility calculation, and we used real-time ultrasound scan for carcass characteristic measurements. So this is diet formulation. We used 13 to 15% of corn germ meal, corn DDGS, and wheat middling to provide enough substrate to Xylanase and Phytase. And positive and negative control diets has about 100 kilocalorie metabolites of energy difference, okay, created by cornstarch and grease in the diet. And then we measured gross performance, average daily gain, feed intake, and feed efficiency. And also we measured back fat thickness and longissimus muscle area, carcass lean percent, carcass daily gain, and lean gain to feed ratio. And we measured apparent total tract digestibility of dry matter, gross energy, ether extract, crude protein, and fiber, calcium, and phosphorus. So firstly, in overall gross performance, we found significant difference between positive control and negative control diets in average daily gain and feed efficiency. So it is an evidence that our diet mixing, okay, and then energy difference were correct. And then firstly, when you look at Xylanase supplementation effect on overall growth, there was no significant Xylanase effect in average daily gain, feed intake, and gain to feed ratio, but there was numerical improvement here in average daily gain and feed efficiency by Xylanase supplementation. And then there was no Xylanase effect on carcass characteristics, then no effect on apparent total tract digestibility of nutrients. However, when you use phytase up to 2000 FTU, overall gross performance, we found quadratic increase of average daily gain, so that 1000 FTU showed highest average daily gain in overall period. And then the feed intake is also increased, but numerically, and gain feed ratio, feed efficiency increased linearly with increasing phytase supplementation level. And also carcass characteristics, carcass lean percent increased linearly with increasing phytase supplementation level up to 2000 FTU, which is super dose. Lean gain as well, and lean gain to feed ratio, okay, increased with increasing phytase supplementation level. And then back fat thickness, okay, decreased linearly with increasing phytase supplementation level. So superdosing phytase supplementation could increase leanness, okay, and reduce back fat thickness and lean deposition in growing and finishing pigs. And then on the digestibility, so clearly phytase supplementation increased phosphorus digestibility, and then it's maximized between 1000 to 2000 FTU per kilogram diet. And then the other nutrient digestibility, so neutral detergent, fiber, and hemicellulose digestibility quadratically increased. Okay, and then ether extract digestibility is also increased with increasing phytase supplementation level, so that dry matter digestibility is increased. So which means phytase supplementation up to superdose level increased not only phosphorus digestibility, but also fiber and ether extract digestibility. So we confirmed this extra phosphoric effect, okay, in increasing nutrient digestibility. And we could not find any interaction between xylanase and phytase supplementation in our measurement. But even though there was no statistically significant interaction uh, in this study, we could find a potential interaction between these two enzymes. So in this graph showed uh, gain to feed ratio improvement, okay, when phytase is supplemented at 500 and 1000 FTU. Okay, so xylanase supplementation could increase feed efficiency with phytase supplementation, but it's numerical difference, so we need a further study to demonstrate this interaction. So summary of this growing and finishing pig study. So Xylanase supplementation to high fiber diet actually had no effect on gross carcass characteristics and digestibility, but there is the other studies having beneficial effect of xylanase on this growth and digestibility. So it may be due to some difference in experimental setting. And increasing phytase level enhanced the growth rate, feed efficiency, and carcass leanness, and apparent total direct digestibility of or different nutrients, not only phosphorus, but also ether extract and fiber, etc. And there, even though there was no statistically significant interaction observed between xylanase and phytase, but there could be a potential, okay, of interaction between these two enzymes in feed efficiency. So in conclusion of this seminar, so phytase supplementation clearly increased the phosphorus digestibility and availability in sows and pigs. And then we also demonstrated extra phosphoric effect of phytase in sows with increased calcium digestibility and milk phosphorus content, and increased weaning weight of piglets and bone integrity of these piglets, and reduced weaning to estrus interval, and in growing and finishing pigs with improved growth rate, feed efficiency, and carcass leanness, and improved fiber and fat digestibility, along with phosphorus digestibility. And then we also wanna do more research about potential interaction with xylanase enzyme. Okay, so this is all I have for this seminar regarding extra phosphoric effect of phytase in swine diet beyond the phosphorus release. And thanks for your attention for a long time in this presentation. And if you have any question, feel free to ask me anytime through the email here. And thank you and bye-bye.